Ladies and gentlemen, daragie друзья, I just came back from Moscow from conference. It means I don't need translator. I'm working simultaneously. I'm sitting on 2 chairs, which is not recommendable, but I work for university and also for big pharma company. And we used to say, if you are not able to explain your grandma in 5 minutes what you are doing, better stop it. It means don't worry. Some people hesitated to come to this seminar. It means don't be afraid. It will be understandable. And please ask any question anytime if you do not understand or you don't agree. Because clearly this is completely new area. Nobody knows exactly what is going on. Many questions are open, but we would like to start with some basics, and then basically Heidi and I, we agreed that it will be done structured way. Firstly, we will describe basic features of genetics, then go to epigenetics and also influence of food, nutrigenetics, and then Heidi will finish with some special topic, which I don't understand at all, but I hope to learn something. It means this will be program for this afternoon, and I skip what I am doing. Basically, let me start with this description. All information is hidden in our cell, and in nuclei there is complete information for us as humans. It's genius design how this information is stored. Just 4 letters, nothing more. 4 letters which are basically assembled as a duplex of DNA, and this is how information is translated into cell. Basic molecule is DNA. It gives complete information about whole humans. Then if we need to translate this information, this basic law of molecular biology creates from DNA, from duplex is basically unwinded to a single strand, and then messenger RNA is formed, and then it gives information for peptides. A reason why we are describing it, because it relates to epigenetics also, because you can have whole information available, but question is if this information is used and translated, and which way is used in special tissue. It means this is quite important to know. Basically, this about 10 years ago was finished the biggest project, the biggest scientific project in a couple of centuries. Some people say it was the biggest discovery after Albert Einstein theory of relativity, because what we know right now is exact sequence. We know exactly what are 3 billions of these letters of DNA in our cells, and it takes many years to discover it. It was quite a tough project. Project was called Human Genome Project, and basically the goal was to analyze whole this sequence and understand how this information is translated. You can ask who was model. Model is average person. As we are sitting here, look how different we are. And we are different in this letters only 0.1%. It means we are 99.9 the same, but look how we are different. It means 0.1% is sufficient to give this huge variety, and it's a very interesting way. This is description how this information is transmitted, because we need, when cells divide, we need single strand, exact copy. And this Human Genome Project basically gives us many information and open many questions. We believe that once the project is finished, it was finished in 2002, that it will basically solve nearly all questions, but many questions were opened. It started 1990. and it was done simultaneously by academic institution and private company. There was even attempt to patent some structures, but fortunately enough, patent of human genome was not allowed. And oops, somehow stopped moving. It means basic information is we know 3 billion letters. We know exactly how adenine, thymine, guanosine, etc. It means we know exact information. We know number of genes. We are nearly the same, but we are very different, and it also gives information about disease, health, and so on. There are some challenges which we have to solve. We can discuss it later on. And what are anticipated benefits? All medicine is now so-called personalized medicine, because basically pharmaceutical companies are developing medicine for average man, but there is nothing like average man or woman. Yeah? We are very different. It means we would like to use this information for improving of diagnosis. Very important is early detection and also predisposition for certain disease. There is hope there will be rational design for new drugs. There is open question for gene therapy. And I had one slide which I couldn't find. Basically, in future, imagine is that you will bring your sequence, your genome, to pharmacist, give it to him, and he will put it to computer, and computer will prescribe you exact medicine. This is basically imagination, because this human genome project costed billions of dollars. Now this sequencing cost something like your flat TV screen, and very often it's believed in a couple of years, price for complete sequence will be like 500 euros. It means affordable for everybody, and you will exactly know what is behind. You will know your risk, and you will know also your options for treatment. Here are two guys who were responsible for this human genome project, and basically I will stop here. And basically this figure shows that whole situation is even more complex, that we cannot explain everything with DNA only. As we have heard in the morning from Heidi, this term epi means above. It means there is even some control mechanisms which goes beyond this, because in the past we believed everything is our genes, and this is our responsibility, but right now we see that it's even more complicated. Therefore, we will spend this afternoon with epigenetics and nutrigenetics.
So are there any questions up to now so that we can answer them before we go on? Just maybe to clarify and use the same analogy I used this morning, you saw this DNA in the genome, and then Professor Kral mentioned RNA and protein. So think about of this as a recipe book that you got from your parents, and you can decide what will I cook today. And think about maybe getting a very expensive recipe book. There's actually one being sold for like 1,000 euros in the market now from a gourmet cook. So you wouldn't want to take that recipe book to your kitchen where you spill oil and flour on top of it. So what would you do? You would copy the recipe on a piece of paper and would take that to the kitchen to cook. Yeah? That's the DNA is the cookbook. You don't want to ruin it, so you copy it in a piece of paper. That's the RNA, which gets destroyed really fast when you don't need it. And then with the little piece of paper, you can cook something. So you know, okay, now I have to get the 2 cups of flour and 1 cup of oil and 2 eggs and whatever, yeah? And that's the protein, so that you have a picture in your mind of this thing. And the central dogma of genetics for many years said the definition of a gene is 1 gene is 1 protein. So actually you defined a gene as something that encoded for 1 recipe. But right now we know that 1 gene on average encodes, or you can make 4 different types of food from 1 recipe, yeah? So I hope now, I don't know what happened. Let's see, I had to change. We'll see. Anyways, yeah, there it is. Okay, so I don't see anything now, but. Okay, well, let me just move so that I can see it. So, I think it's important for all of us, even if we're nurses or not even working in the health field, to understand something about genetics. Because, like it was already mentioned, in a couple of years, I mean, right now you can go and get your whole genome sequenced and interpreted and get a free iPad for 5,000 euros. You can do that in London this coming autumn. Yeah. And I think in 2 years' time, it will cost $1,000 and not more. Yeah, so it's going to change medicine. And I think I liked this definition that I found: superior doctors prevent the disease, mediocre doctors treat the disease before evident, and inferior doctors treat the full-blown disease. I hope that many of us can help things, model things so that we can get to the top definition of doctor, yeah? One who can prevent. And I don't want to say that every disease is already written in your genes, but for some, you can already find if you have a predisposition or if you're in the direction of going to develop something to prevent it before it happens, yeah? So I think many are looking like this nowadays. It's a big challenge, I agree, yeah, to keep up with all the developments. But let's take it easy. And this was a quote that I found from Bill Gates. I mean, he's in computer and business. But he said he gave a lot of money now in January, I think, and he says there, advances in understanding the human genome are having a dramatic impact on almost every area of medicine, going from dermatology to orthopedy. I mean, in all disciplines. Some doctors are incorporating it more. The majority, I guess, is not incorporating it yet, but there is nowadays enough knowledge to incorporate in all fields. And this is a comment made 2 weeks ago by the editor of the British Medical Journal, and he says, we are all going to have to learn about genomic medicine, whether we're a clinician in a specialty, or if we're doing something else, or if we're even a layperson. Personalized medicine is the future. Let's get our heads around it and help others to do the same. So we hope that we can be a help today for you and that you will be able to help other people. So there was I think it's the 25th of April, it's DNA Day, and a company called 23andMe carried out a survey, and they asked people in the United States what they knew about DNA and so on. And half of the women thought that they had an XY chromosome. And 1/4 of the men thought they were XX. So men know more than women, apparently, at least what's regarding genetics. The interesting thing is, if you look on the right side, that many people, I mean, there's no stopping to this. There's no saying, no, it won't change medicine. Many people asked if they would agree to have, or they would want to get their genome sequences, are saying they want, 71%, yeah? So we have to learn about this. And on the left side, you can see that's a Canadian survey. On the right upper hand, you can see how many people think they would get their genome sequence and think it will help us understand better disease. So you see, people, the general public is going to be interested in this. And it's happening in the United States that patients are coming to the doctor and saying, here is the result of my 23andMe test. So what do you think, doctor? And many times the patient knows maybe more than the doctor. So start with time getting, information about this. So we already, is this working? Yeah, okay. We already saw the cell and everything, and I just wanted to share with you some things that came out of the Human Genome Project. It cost a lot of money, but it made it possible to find out that we have, wait, I don't want my computer to. It made it possible to find out that we have around 20,000 genes, and that's much less than what people thought we would have. The reason we have many less genes than what we thought is that in humans, a lot more from these alternative recipes are made. So in other organisms, they have maybe 17,000, 18,000 genes, but it's mostly then that same number of proteins. In humans, we have a lot of different, you get the same recipe, and maybe instead of using sugar, you use honey. Or instead of using wheat flour, you use whatever, rice flour. And then your product, what you're going to eat, tastes a little bit different. The same thing is with the genes. You have the same information, but you can just change some things with the ingredients so that you get a different product. So right now it's estimated that we have around 80 to 100,000 different proteins needed for the functioning of our body. And this is the information that's here. The interesting thing is that we have around 12,000 genes, which are, so the sequence is read, but never a recipe is made out of these genes. They are called the non-coding genes. So they're copied, but nothing is made out of them. And we're just beginning to understand what these genes do. They have an important function, but no protein is made out of these genes, yeah? 12,000 of them. Then we have small ones that are not translated either. And these mRNAs, they are very small. They're only about 25 letters long. But they're the third epigenetic mechanism. So today I talked about two mechanisms. This is the third one, miRNA. And what they do is they're very short and they're complementary. So they match. So you know when you take, what is it called? Magnet. When you take a magnet and it attracts itself, well, with the DNA, you have two strands, and when they are complementary to each other, they just get together like magnet, positive and negative pole. And the mRNAs, these 25 base long pieces, when they find a long sequence of RNA, which is complementary to them, they just go and stick to it. And when they stick to this long sequence, another protein comes and destroys the whole thing. So mRNAs are there to, let's say, kill, or let's say you were planning to cook something that your kids don't like. And they come into the kitchen and they say, oh, mom is going to cook, I don't know what, this evening. I don't like that. I'm just destroying the recipe. I don't want to eat that this evening. The mRNAs do exactly that, okay? And that's going to be used, probably, it's already being used for therapy, because it's easy to get these miRNAs into the cells and target specific genes that are doing something bad in the cell, and you don't want them, yeah? So just so that you have an idea what the terms that you can see here mean. So we're talking about genome, and we have to think there's 4 types of different DNA. So we have the autosomes. So there's 22 chromosomes numbered from 1 to 22 that both males and females have in the same quantity. Then we have DNA on the either X or Y chromosome, which I will correct the survey from the US. Women have 2 X chromosomes and men have 1 X and 1 Y chromosome. And then very ignored is the mitochondrial DNA. So we have little things inside the cell, outside of the nucleus of this downtown region of the gene, of the cell. You have, in the periphery of the cell, you have the mitochondria. And inside of the mitochondria, there is also a genome containing instructions that are not found in the nucleus. And the interesting thing about this is that the mitochondrial DNA is passed only from the woman, to both her children, to son and daughter. So the man never passes mitochondrial DNA to his children. So actually, children get a little bit more information from their mothers than from their fathers. So I guess I already talked about this, so I'm skipping this. You will get this if you sign up for it. So this mitochondrial genome contains 27 genes. And has anyone heard about a controversial therapy being carried out in the UK with a transfer? No? Okay, then I won't get into it. But it's, when you know the background, it's not as terrible as it's being talked about in the internet. So we come back to the genes. On average, one gene, so one recipe, if you had to type it, it's 3,000 letters long. That's the average length. There's one that's really big, yeah? And we know the function, so we know what you can cook and how that recipe tastes and where you can find the recipe, only for about 50% of the genes. So there is still a lot of work to be done. Chromosome 1 has the most genes, Y has the least genes, because women can do without a chromosome Y. So the other thing is that genes have a structure, and I wanted to do a little small exercise for this. So I need some volunteers to come up. You don't have to say anything, you don't have to speak, you just have to stand up. So about 10 volunteers. Okay, so this is what you read when you copy the DNA. I mean, it's a little bit difficult to make sense out of it, yeah? So let me see if I can see it. If I were to read that is, that is, hat is, not is, hat, it is, yeah? It doesn't make much sense. So actually, you have like to organize this information. And now I will go and try to make a little bit sense of this, okay? Just have a little bit of patience. Marks, and you need to separate things to be able to understand it. So, not is not. So we need a dot here. So separate your hands. Is that
8? Yeah. 8.
Is that it?
Yes.
It is.
Yeah.
So I think I missed some punctuation marks, but you see, when you organize the information, then it makes more sense. When you copy the DNA, you're copying just a bunch of letters, and you have to break them up and put some punctuation marks so that it will make some sense. That is, that is not, is not. Is that it? It is. Yeah, but at the beginning it was hard to comprehend when you don't have these punctuation marks and breaks between. Thank you all. You can take your papers and throw them away. Okay. So that's exactly what happens when you copy this whole information from this recipe book. It's a little strange recipe book. It has things in between which seem not to make sense. And then when you copy it to the RNA, you have to cut it out and put it together again so that it would make sense. Why? Why should that be? Well, that gives the cell the chance to make different things out of it, because it can leave out one word. I could have taken out maybe the middle sentence of this phrase now, and the meaning would have changed, yeah? So that's what you do when you do this different splicing. And then you have the genetic code is read in 3, so there's a question?
In this long DNA chain, do the pieces change from one place to another?
No, they cannot jump by themselves. So
you have done this?
No, you do that in the RNA.
Okay.
So the DNA, the recipe book that you bought for $1,000 stays always the same. You just copy it in a piece of paper and say, today I want to do it like this. I will put more sugar, so I'll keep that, or I'll leave the sugar out, and so on. So the genetic code is read in 3-letter words, so to say. And each 3 letters tell the cell, okay, now you have to get an amino acid called phenylalanine. That's the first one on the left. Or you have to get a serine. That's the second one, and so on. And you see for some amino acids, it doesn't, it's the same if you have a CCU, a CCC, or a CCA. They're all telling the cell, go get a proline, bring me a proline, bring me sugar, yeah? If you're a little bit mixed up, why now we have a U? That's because on the RNA, instead of a thymine, you have an uridine, yeah? But don't get mixed up with it. Just think about 4 letters, it's the same. So, any questions about how the genes encode information and how this is read before we go on? Okay, so we're going on. Okay. So what types of mutations do we have? Because that's what, most people talk about, mutations. So there's the big mutations where you have one chromosome too much, or you're missing part of a chromosome. And then you have the point mutations, where it's just 1 letter. So here is an example of different chromosome things that can happen. And we all have these things, yeah? Each one of us carries some type of these big changes on the chromosome. We're missing maybe 1 gene, maybe a gene that's not so important, or we have 2 copies of a gene, or it's turned around, yeah? So that's, let's say, normal in human beings nowadays. It wasn't in Adam, but it's normal nowadays. So each one of us has about 1,000 errors, big errors of at least 2,000 letters in our chromosome. So nobody's perfect, yeah? So mutations. What you usually think about when you hear mutations is about a change in 1 letter. So if everyone has a C, you have a G, yeah? The difficult thing is to see if that G is really a mutation, or you're just a little bit different and it doesn't matter. It may not matter, yeah? So how do mutations happen? One way is that when you're copying the DNA, each time a cell has to divide, it has to copy the whole genome. Yeah, it takes about 24 hours to copy the whole 2 meter long sequence. Of course, if you're smoking or you're taking sun, you're exposed to mutagenic agents, and they lead to changes. For example, do you know what happens when you are exposed to UV light? What happens is that instead of the 2 strands staying together like they should on that little picture on the left, 2 bases that are besides each other decide to get together and separate from the other strand. And then a lot of enzymes have to come and break that up and repair it so that you don't get any mutation from being exposed to sunlight. I think when you're exposed 1 hour to sunlight, about 17,000 of these things happen in your cells. Yeah, and they all have to get repaired again. So, and then there's spontaneous changes. So to explain mutations, maybe this is the best example, and I left a paper on every second seat where there's examples of how mutations, what their effect is. For example, let's say here I used all 3-letter words, yeah? So the big dog has 1 red eye. Maybe instead of having that G in Verena type, you have that G in whatever, yeah? You change the letter, but it doesn't change the meaning of the word. Yeah, that's called a silent mutation, something that doesn't have any effect on the recipe. Then it could be that you change the letter. The bean dog has 1 red eye. It doesn't make much more sense anymore. That could be more relevant for the protein. Or it could be that you introduce a stop. For example, the big, stop. The sentence hasn't been finished. And sometimes that happens. So you begin cooking your recipe and you put the first 2 ingredients and put it in the oven. Is it going to taste like the real thing? No. That happens a lot of times with mutations. The protein gets started and then it's stopped. And then what can happen is that an extra letter comes in the middle, the big O, do, ga, son, ere, de, e. So the same letters are still there, but if I read them in 3 blocks, then it doesn't make any sense after big, yeah? And then some letter might go missing, like the G and the big O, a, son, ere, de, ye, yeah? So that's the type of mutations that can take place. Mutations that have no effect, mutations that lead to a change in the meaning, mutations that lead to a stop, or to something that's meaningless. This is illustrated here, and this is what you have in your seats. So this is the same. I think I will skip them. So mutations, how do they arise? You've all heard that maternal age is really important. You know, women older than 35 have a higher risk of getting a Down syndrome kid. But did you know that the newest information says that fathers, when they're older, they also pass on a lot of more mutations than when they're young. And nobody tells you this. And no gynecologist is going to ask how old is the father of this child that is going to be born. But fathers pass 4 times as many new mutations as mothers. Yeah, it's different. Mothers pass like big mutations, like Down syndrome. Fathers pass on small mutations, but that on the long run may have an effect. Yeah, and like the number of mutations that a 70-year-old father passes on to his child is 8 times more than the 20-year-old father. So also men take this into account. You know, you can have children even when you're old, but they will carry more mutations. So what came out of the human genome? Nobody's perfect, I said. So each one of us has about 3 million differences to the one sitting behind you. Yeah? So 3 million letters are different. And about 200,000 of those are changes that you will only find in you and in nobody else in this room. Yeah? So those are called the rare or private variants. And each one of us carries 22 pathogenic alleles, which may lead to a recessive disease. So each one of us carries, let's say, a faulty gene for a disease. The good thing is that for this type of diseases, you need that both the mother and the father carry the same mutation, not the same mutation, but a mutation on the same gene for their child to be sick, yeah? So if you have luck, you marry someone who doesn't carry mutations in the same 22 genes that you carry, and you have healthy children. Just that you get a picture, nobody's perfect, and we all carry mutations. The thing is the mutations that I carry, do they have any relevance or not? Yeah? And also carry about 300 predicted loss of function variants. So this type of variants that put in a stop in the middle of the sentence, for example. Yeah. So how do we define a mutation? I told you we're all different. There is a definition that is used, but I mean, we still don't know how to define what is a mutation and what is something that's just a change, but it doesn't have any relevance. So usually people say, if it's present, if you can find the same change in more than 1% of the same ethnical group, then it's not a mutation, it's called a single nucleotide variant, which is also called SNP, yeah? And maybe it's not even in the gene, but on the regions that don't contain any recipe. If it's in less than 1% of the population, it may be a mutation. And it's usually present in the recipe itself, yeah? So mutations can be good. Did you know that? There are some mutations that provide you an advantage. So, for example, people carrying 2 copies of the mutation in the CCR5 gene, that's the 4th one, they never get AIDS. Even if they come into contact with a HIV virus 20 times, they will never get AIDS. Why? Because CCR5 encodes for a protein that's on the outside of the immune cells, and the HIV virus needs to find this protein to be able to get inside the cell. So if the people can't make a faulty receptor, the virus never can get inside any cell, and it just dies. So not all mutations are bad. There's mutations that give you extra strong bones or low coronary diseases, and there's even, so this is interesting. The picture here in the bottom is from Timothy Brown, I think. He had AIDS. I mean, he was sick with AIDS, yeah? He had been infected, had AIDS, but then he got leukemia. And I don't know how, but they decided to do bone marrow transplant on him. And he got new bone marrow to treat his leukemia. And the positive thing was the bone marrow that he got transplanted had the CCR5 mutation. So he got cured from AIDS. And now he doesn't have AIDS anymore because he got new immune cells that don't have this. Yeah? So don't think, I mean, you cannot say mutations are bad. For most things they are bad, but sometimes you have an advantage when you have a mutation. Yeah?
Why don't they give the same bone marrow to all the HIV positive ones?
It would be maybe something to do, but I guess it's difficult to get so much bone marrow for all AIDS patients. But I mean, yeah.
And it's a risky thing to get the bone marrow transplant. You can die.
Yeah, of course.
To match many other factors.
Yeah. So other mutations that have been found, for example, there is a mutation in the GHR gene that if you have that mutation, you will be protected, so to say, from cancer. And it will reduce your risk of diabetes also. There's different examples. So mutations, for example, in the ApoC3 gene will reduce your risk for hypertension and diabetes, and so on. So mutations can have a positive or a negative effect. And this is just summarizing what I talked about. The other thing to remember is that not every person carrying the same mutation will get sick. Yeah? There's a lot of factors playing a role. So even if 2 siblings get the same mutation, brother and sister or 2 brothers, it may be that one has symptoms and the other one doesn't. That is called a variable penetrance, yeah? Or one, let's say, may have more severe symptoms and the other one doesn't have so much problems. That's a variable expressivity, oh, well, whatever, you know. So there's it's not just the one mutation causing disease. The other genes and other lifestyle factors will have an influence on how it is expressed. There's genes and mutations that, like 99.9% of the time, lead to a disease, like Huntington disease, yeah? But there's other ones where you cannot really predict what will happen. So it's not so easy to find what causes what. And this is the moment in science where we are. Right now, many of the so-called Mendelian mutations have been identified. Things for Huntington and cystic fibrosis and breast cancer dominant, have been found and so on. But then most of us will get, I don't know, hypertension, diabetes, things that are very common, but where you don't have one punctual gene that leads to that disease. So this is like this right down corner. And this is where we are now, trying to find out, and you need a lot of patients and get their information from their genes and from what they have to find out, okay, this is the connection, this gene with this disease and this gene with this disease. I will skip this. Inheritance. I think you all learned in school. So does someone want to go over this, or I'll skip it? Skip it, okay. I'll skip it. I'll skip this too. So I talked about the SNPs, yeah, the single nucleotide polymorphisms. Usually they're not inside the gene and they will not affect the protein, but they may affect the quantity of food or gene that is expressed, yeah? So most of these diseases that affect many people are due to SNPs. And this is what many studies called GWAS, genome-wide association studies, which I showed before, are trying to define. So we have on the one side what's called the genotype. That's the sequence of letters that you have on your genome. And on the other side, you have what, am I healthy, what disease do I have, and so on. And right now, we're trying to make the connection. So how do you find out the sequence of the letters? Is that easy? Is that complicated? For many years, for like 25 years, Sanger sequencing was used. Then in the 90s came another new technology called Determinator. And nowadays, we're using the so-called next generation sequencing. And this has allowed us to go from that, which was radioactive sequencing, to something, you see what that is here on the top? It's a pen drive. It's something you can put in your computer. And there's a company that has already made a pen drive that you can stick into your computer. And there's a little hole. You just put a little bit of material, usually right now for bacteria, and then it gets sequenced. So you just plug it into your computer, and it gets electricity from the computer, and there's a whole circuit inside of this little pen drive, and it does everything that has to be done, and 2 hours later, in your screen, you see the sequence of the bacteria that you put in there. This is being applied. This is not science fiction, yeah? So just that you know the advances that are taking place. It's advanced, I mean, you cannot have any idea how much it's advanced. You see on the top side, you see the cost of sequencing a genome and how it's developed in the last 15 years, and it's dropped amazingly. I mean, maybe someone's familiar with Moore's law. Moore's law says that so knowledge duplicates, or the computer power duplicates every, I don't know what it is, 1 year or something. I mean, and that's Moore's law, the white line, and so these advances are even faster than Moore's law. So right now it takes 3 hours to sequence a complete human genome, and it costs between $3,000 to $5,000 depending on the company. So what can you use it for? You can use it to identify genetic variants for diseases that are inherited in a Mendelian way. You can do newborn screening. You can do prenatal diagnosis and do preconceptional testing to see if the parents carry both the same mutation in the same gene, for example. I know this all may sound like science fiction. This is a nice illustration from the year 2011. It shows what's going to be taking place when. So now we're in the third lane, so to say. The red shows you what's happening where we are now, and then it shows how it's going to be expanding. So right now we're still in the understanding of the biology of the genomes and the biology of disease. And in about 7 years, it will be really being applied most of the time to medicine if things keep on going like they are going until now.